Okay, so hello everyone. This is the first webinar I've given, so just apologies in advance if anything goes wrong, but hopefully it will go smoothly. So the title of my talk is Genomic Studies of Neurodevelopmental Disorders and the Move from Arrays to Sequencing. But right, okay. So before I start talking about some of our results, just a bit of background about us. We're a research lab, and our sort of main goals are firstly to identify novel disease genes, but also to sort of try and translate research findings into sort of clinical practice. So we've got very strong links to a number of clinical labs and clinical geneticists. Okay, so sticking with my title, I thought I'd present 3 different cases, which have all been published now, but they sort of highlight this sort of gradual sort of transition, firstly from using, the first case is talking about, for the first case we analysed with arrays and Sanger sequencing. The second case I want to talk about, we used arrays, a combination of arrays and exome sequencing. So that's more sort of the current sort of state of play. And then the third case we went straight for whole genome sequencing. But all these cases, I think, have some interest all in their own right as well. So I thought it made a nice mixture. And then at the end, I've got a few sort of technical slides if people are still listening at that point. So the first case, the patient had a structurally normal but a small microcephalic brain. They also, as you can see from the pictures, were, they had sensorineural hearing loss from 3 years. They were screened for mutations in the most likely candidate gene, which was ASPM, and they were also checked for CMVs. So there was 5 unaffected SIBs, and there was no family history, so we thought initially the most likely mechanism was a de novo mutation. But we didn't find any CMVs of clinical relevance that we would want to go back and test parents. However, because we were using SNP arrays, we were able to see 2 large stretches of copy-neutral LOH involving the short arm of chromosome 7 and the long arm of chromosome 9, which is shown at the bottom here. On the left, chromosome 7, and on the right, chromosome 9. So initially we thought this was strange because the parents were meant to be non-consanguineous, but we added up the sizes of these 2 regions and worked out the coefficient of inbreeding, which suggested that the parents may have been something like second cousins, or second cousins once removed. So we went back to the clinician at that point, and they sort of sent us this pedigree. So we were correct in that this confirmed that we were right, the parents were second cousins. So at this point, I think typically we might have thought about exome sequencing to sort of look at all the genes in this region, but we noticed that one was a really outstanding candidate gene, and it was a known microcephaly gene called CDK5-RAP2. However, at the time this was one of the sort of rarest microcephaly disease genes, and in HGMD in 2012 there was only 2 mutations reported in this disease gene. So because it was such a good candidate, we thought we'd save money and just do Sanger sequencing, so we sequenced all 38 exons, and we found a novel transversion mutation that predicted a stop codon. And so at the time this was only the 3rd mutation to be described in this gene, and it was the first mutation described in a patient who wasn't originally from or descended from Pakistan ethnicity. So this slide is now just showing the Nexus software. We've loaded in the SNP array data, and the orange is just showing the LOH call, and then we've also loaded in sort of the position of our coding variants that we found. So there was 2 known coding SNPs, and then our novel stop mutation. So that's all the data from the first case. So this showed a number of things. Firstly, we were able to correctly infer parental relationships. Obviously, in some situations this can be sensitive. For example, if you see larger coefficients of inbreeding, and there's papers on this. It also helps confirm this CDK5-RAP2 as a rare microcephaly gene, especially because it was the first patient described outside Pakistan ethnicity. We were also quite lucky. Initially, we were going to run ACGH on this patient, and we only ran SNP because we didn't have enough DNA, so that turned out to be quite lucky. But I suppose these results highlight the advantage of SNP over ACGH, especially in cases where there's any possibility that there might be some consanguinity. And so it's invaluable as a pre-screening tool before embarking on expensive genome and exome sequencing studies. But I suppose as the cost of whole genome sequencing and exome sequencing decreases, this last point won't become so important anymore moving forward. So the second case, this was a Pakistani family this time, comprising 3 affected siblings. And this time the parents, again, were second cousins, but we were told that right from the start. The patients all had developmental delay, learning disability, they weren't able to speak, they had psychomotor delay and also postnatal microcephaly. And again, ASPM and CMVs had been excluded. So again, we ran, because we realized the parents were related, so we wanted again to look for regions of autozygosity, and we ran cytosnip12, so Illumina SNP array, and we found a single autozygous region on chromosome 17, and this is just showing that data. It's a little bit fuzzy, but I've sort of highlighted the region that's shared. You see there's no heterozygous SNPs there. So that was our sort of candidate region. So then we exome sequenced just the proband, so one of the 3 kids, and we got a list of about 200,000 variants, of which 191,000 were high confidence, and 11,000 were rare, and of these 144 were predicted deleterious. And then because we were looking for a homozygous mutation, the genetic analysis was just looking for where both alleles have the variant. And then we've got 12, but only one of these was in our region on chromosome 17, and that was a missense change in this gene called PGAP3. So we went back, the Sanger sequencing confirmed that all 3 affected children were homozygous for this variant. I mean, this was already expected because of the SNP array results, but also the parents were both carriers. So this was consistent with autosomal recessive inheritance. So to follow this up, we sort of checked the amino acid conservation at this site, and it was conserved right across all the species that we looked at. It wasn't in the exome variant server database. It wasn't in 1,000 genomes or in some in-house genomes of mixed ancestry. But as this family were from Pakistan, we also managed to get hold of 108 Punjabi controls, so very close to where this family originated from, and they were also not. We didn't find this mutation in those individuals. So what was interesting at that point, we found that there's been some biochemical testing done, and all 3 patients, all 3 kids in this family had elevated serum alkaline phosphatase, and this is actually quite a good marker for defects in the late stages of GPI anchor biosynthesis. And this PGAP3, that's exactly the pathway that this gene is involved with. So at that point, I think that was the time where I sort of really thought this was the gene that was actually causing the disease. We also collaborated with a group in Osaka, Yoshiko and Taro, and they carried out some FACS analysis on some CHO cell lines, and they confirmed that the missense change that we'd identified did result in a functional loss of activity. So you can see the black line is moving from the right to the left when you add the wild type cDNA, but when they're adding our mutation, it stays on the black line stays on the right hand side. We also collaborated with a group in Berlin who'd had pretty much at the same time identified 2 other families with mutations, biallelic mutations in this gene, and pretty similar phenotypes, and we all wrote this data up together, which was published in January this year. So that was the second case finished. So just a background before I go on to this third patient I want to talk about. So the third patient came from this WGS 500 study, which is a collaboration between the Wellcome Trust Centre for Human Genetics, where I work, and the Oxford Biomedical Research Centre, and also with Illumina. The project started in 2010, and proposals were invited from clinicians all over Oxford, and we wanted to try and recruit cases where standard genetic tests had been performed but had been negative, or else conditions where there was no test available. So the aim was to try and recruit a sort of quite a diverse set of phenotypes, and we did quite well in that regard. We've got over 40 different phenotypes, and we wanted to, with this patient cohort, we wanted to look at the utility of whole genome sequencing. So we had a mix of Mendelian or presumed Mendelian single gene disorders. We also had some patients with immune phenotypes, which we thought would probably be more complex, and then we also had some cancer samples. So there's been a bit of coverage in the media on this project, but also it's been quite successful, and there's been 8 scientific papers already. There's been, so some of the other sort of headline results are that there's been 7 novel disease genes, 6 novel phenotypes for known genes. There's also been 2 regulatory mutations, either in UTR regions or downstream of known candidate genes, and these are sort of good examples of where whole genome sequencing has advantages over other methods. There's also like about 2% of families we've seen have variants that were previously should have been picked up by Sanger sequencing but somehow were missed. And then in total of almost 100 presumed Mendelian families, there's been 23 new clinical diagnoses. So the figure at the bottom is just showing a sort of handful of these phenotypes that we recruited, and the ones I wanted to focus on here, we had 7 patients recruited with either with Ohtahara Syndrome or other sort of early onset epilepsy. So these were all severe early onset forms of epilepsy, and this figure is just sort of showing sort of how well we did, if you like, across these 7 families. So only 1 family we didn't have any candidates. 2 families we've got reasonable candidates but we're not completely sure about. We've got 2 families where there were de novo mutations in sort of pretty well established epilepsy genes. 1 patient where there was a homozygous splice mutation in a gene that acts further down this GPI anchor pathway. So that was our sort of novel, that was a novel gene for this epilepsy disorder. But the one I wanted to focus on today is actually a known epilepsy gene, but it was previously described for a different type of epilepsy. But it's, as you'll see, it's also quite interesting on a number of levels. So this was a patient with Ohtahara Syndrome. From the age of 2 weeks, they had 40 seizures per day, severe developmental delay, and also the metopic suture had fused prematurely. So the EEG test showed that they had this peculiar electrical sort of suppression pattern, which is diagnostic of Ohtahara Syndrome. There was no family history of this type of epilepsy, but there was other family history, a paternal great-grandmother and a great-aunt who were reported to have a milder form of epilepsy. So with conventional methods, like over a period of 3 years, there was 5 different genetic tests carried out, like costing about €4,000, and all of these were negative. And with conventional testing, they're performed sort of one after the other. So if we'd sort of carried on with this route, I mean, we wouldn't really have, I don't think we'd have found the causative gene for a very long time. So anyway, at this point, this was one of our first whole genome sequencing studies to come off the machine. I started looking at, so I hadn't looked at whole genome sequence data ever before, and I'm not really a bioinformatician. I'm more of a sort of lab-based scientist. So I started sort of just sort of eyeballing some of the data just to see what it looked like, sort of picking out a few of the sort of candidates, early onset epilepsy genes, and a few of these happen to be on chromosome 9. Now this is a slide I've used for other presentations, and at this point this is my sort of audience participation slides, if you like, which obviously doesn't work quite as well with on doing a webinar format. But so hopefully people who are familiar with using IGV and looking at illuminated data will spot straight away what the problem is here. So the patient's in the middle and the parents are on either side. So you can see on the right there's a homozygous variant in the patient that's only present in the dad, and on the left there's another Mendelian error, namely that the mother's homozygous, but neither of those chromosomes seem to be transmitted to the patient. So at first I thought maybe there's sort of a deletion being transmitted from the mother, but there doesn't seem to be a drop in sort of copy number. And then I just sort of looked all along the chromosome, and this pattern sort of carried on. So we did a more formal sort of analysis of this. So we plotted the ratio of heterozygous to homozygous variants in 1 megabase windows across this chromosome, and this basically confirmed that the whole chromosome was what we thought was like uniparental isodisomy. So they've got normal copy number, 2 chromosomes, but they're actually just the same chromosome twice. So right away along the chromosome, and these are the parents on either side again for comparison. So we didn't like the way I mean, we thought we could plot this a bit better. So we actually, so as well as the whole genome sequence data, we then wanted to just validate this. So we did go back and do SNP array, and then we've loaded both data forms into Nexus Copy Number. And I mean, we've had to clean the whole genome sequence data a bit, but these are the sort of B allele frequency type plots at the top from whole genome sequence data, and at the bottom from the array. And obviously it's very noticeable the heterozygous cluster in the middle is missing completely right away across the chromosome. And this is just, I think this is chromosome 9, this is chromosome 10, just for comparison. So you sort of miss this heterozygous cluster. So the cytosnip 12 data confirmed the UPD, and there was only a single heterozygous SNP all the way along the chromosome called as heterozygous. So then the next question was, is this actually what's causing the disease? So there was 2 hypotheses. Firstly, it could have been that there's an imprinted gene on this chromosome that's causing the disease, but there's not much literature supporting that hypothesis. So then we thought, okay, well perhaps there's a recessive CMV or recessive point mutation somewhere on this chromosome that, because of the UPD, is suddenly homozygous in the patient, and that's what's causing the disease. Unfortunately, the SNP array data, the log-R ratio was a bit sort of noisy, so it was difficult to sort of see if there was any small CMVs along the chromosome. I mean, there's one that comes up as being called here, but it wasn't very clear. Another way we thought of looking for CMVs along this chromosome was looking at the SNP array data, looking at these unusual, sort of unexpected genotypes. So anything that's coming up with an intermediate B allele frequency is a bit dodgy. I mean, you know it should be homozygous, but it's being called as heterozygous. So we focused on those about a dozen SNPs that had that pattern, and they were coming up with an intermediate B allele frequency, but really they were just really low signal. So I'm showing 2 here examples. This is the clustering plots from the SNP array, and then what that looked like on the B allele frequency plots. So there's one here and one here. So because we had whole genome sequence data, we could then zoom in on those 2 regions, and one had a low signal because there was a variant, like I think 3 base pairs away, so it wasn't genotyping very effectively. But one of the sort of dozen or so SNPs we looked at, it was genotyping. It had a low signal from genotyping because it was a homozygous deletion, and this was the thing we were sort of looking for, these type of CMEs to see if any of them could be relevant for the phenotype. So here we've got about a 3 kilobase deletion that's homozygous in the patient that was inherited from the heterozygous father. So this, we thought this was quite a nice way of sort of mixing sort of array and whole genome data together to sort of find CMVs. However, this deletion didn't hit any genes of interest. However, we still, because it was the first, I mean, we were quite early looking at this, was quite early on in our sort of use of whole genome sequence data, so we wanted to validate that we'd picked this was a real deletion. So we designed some primers specific for the non-deleted chromosome and also specific for the deleted paternally inherited UPD chromosome. So we ran those PCRs, and this enabled us to validate the deletion, and it was actually slightly smaller than 3 kb. This also enabled us to test DNA from a different tissue. So in saliva, there was also no evidence of any maternal chromosome. So we couldn't find any evidence for mosaicism of the UPD. And this next slide is showing, I think it's a figure from a 2010 review showing like different mechanisms that lead to uniparental disomy. And so in this case I'm describing here, for a number of reasons, we think the mosaicism, the UPD occurred due to monosomy rescue, and that's because there wasn't any evidence for mosaicism. It was isodisomy, and that was right across the whole chromosome. So basically what we think's happened is that the sperm was normal, like the sperm was okay, but the egg was originally missing chromosome 9, so the zygote then was initially monosomic for chromosome 9, which would normally be lethal, but then there's been a rescue event, and that daughter cells of that rescue event have sort of outgrown the sort of non-rescued cells. So because we thought it was due to monosomy rescue, I mean, having 2 extremely rare things happen, we could say to the family that there's a very low risk of recurrence of this condition. But we still haven't found what's, so the first CMV, the first deletion I showed didn't intersect with any other genes, so we wanted to check further, are there any other CMVs that we've missed along this chromosome? So we looked closer at the sort of read counts along this chromosome using this NGBin Java-based utility, which was originally based on a CMV-sec paper I've shown here. So we looked, because we were just looking at a single chromosome, we could look very closely, sort of manually, right along the chromosome, and we picked up 4 additional CMVs, so 3 deletions and 1 duplication, which were all homozygous on account of the UPD. However, all were listed in DGV, so I'm just showing these quickly. So here we've got 2 consecutive windows with low signal overlapping. The pink rectangle here is the DGV track. So straight away you can exclude it, and it also doesn't hit any genes. So here's the IGV trace for that one. So it's pretty clear. And then these are the last 2, another deletion, again overlapping with a DGV, a common deletion, and a duplication. So this was 4 copies instead of 2, again overlapping with a known duplication, and again not hitting any disease genes. So although we've shown this method's quite good at picking up, all these CMVs are sort of slightly below sort of SNP array resolution, but unfortunately none of them seem to be what's causing the disease. So then we go to the whole genome sequence data and look at all the point mutations and indels along chromosome 9, and so we found 443 coding variants along this chromosome. 59 of these was seen at 1% or less frequency in the 1000 Genomes project. And once you've got down to sort of that sort of level, you can sort of look through the gene list, and one of them was a sort of exceptional candidate gene, a potassium channel gene, and that was the one that we were pretty sure right from the start, to be honest, was what was causing the disease. So sound sequencing confirmed it was homozygous in the patient and inherited from the dad who was heterozygous. We couldn't unfortunately get DNA from the two more mildly affected paternal relatives, but the paternal, we did get DNA from the dad's mother who had the variant, so that's still consistent that maybe if it's homozygous it can cause this severe epilepsy, and if it's heterozygous maybe it causes a milder form of epilepsy that's not always penetrant. And actually while we were sort of working on this study, there was 2 papers that published dominant mutations in this gene causing a milder form of epilepsy. We've also collaborated with a group at Yale who did electrophysiological studies and showed that the electrical currents, when you've got this mutation, or if you like, the channel opens much more easily and you get higher electrical current. So to summarize this third case, so we think the causative mechanism is paternal UPD leading to homozygosity of a missense mutation not in this KCNT1 gene. The UPD was due to monosomy rescue, so this is useful for the family because they now know it's pretty low recurrence risk for this form of epilepsy, and it extends the phenotype associated with KCNT1 mutations in epilepsy to include Ohtahara syndrome. Also, some nice things about the study was we showed that, I mean, the whole genome, if you'd done this with a sort of previous strategy of doing it array first, you'd have found the UPD and then you'd have had to have at that point decided, oh, do we sequence random genes along chromosome 9 or do we do exome sequencing? It would have been a it would have involved multiple different stages, but using whole genome sequencing you can basically do everything in parallel, so you can find the UPD or the homozygous regions while you're doing the variant calling, and once you've found your candidate region you've got your list of variants straight away to sort of look through, so it's much quicker than other methods. And then another nice thing about the study was just showing that integrating the sort of unexpected variants, unexpected genotypes from the array, integrating those with the WGS data can be informative. And then also the read coverage analysis at the end was, even though we didn't find a causative CMV, it was a good way of picking up CMVs that were below array resolution. So those were the 3 cases I wanted to present that sort of show the sort of move from using arrays as a sort of first pass analysis to going straight for whole genome sequencing. The last few slides I've got are just sort of more technical points of interest, so not really sort of interest in terms of phenotype, but yeah, more sort of technical things. So this slide is showing this is now onto another family where for 3 family members we used SNP arrays, and then for another family member we used whole genome sequencing. But there's 2 tracks at the bottom for the whole genome sequencing data, because so this is plotting or trying to plot log R ratios. So from the 3 SNP arrays, and then we've got this one here is the coverage at specific discrete common SNPs, and then this is the coverage in terms of like how many reads within 5 kb windows. So as you imagine, there's more data that's contributing to this signal, so it's cleaner. But this was interesting. The reason I'm showing this because I guess an advantage of whole genome sequencing is that you can find sort of slightly more complex rearrangement. So if you just look at the log R ratio, you think, okay, it's just a duplication, but it doesn't actually tell you like where that third copy is. It might be somewhere else, it might be inverted, it might be a standard tandem, direct tandem repeat. So the majority of duplications are this sort of direct tandem repeat, where you get read pairs at the edges of the duplicated region that point outwards. But for this one that I've just shown, it was a slightly different pattern. So we're getting 3 read pairs with sort of abnormal sort of configurations. which are sort of shown in this schematic, but also in the real data here, you can see. So what we think, what happened here is that there was you get the extra copy, which has then been inverted and then inserted a few kb downstream, but then we've also had a little bit of the middle had been lost. So that's what these three junction fragments, if you like, that lead to the aberrant read pair mapping, and that's where they come from. This was an 80 kb duplication that was complex and in an inverted orientation, and we've looked at a few of these now, and it seems like sort of about 80% seem to be in the sort of common standard tandem repeat orientation. So it's the other 20% that it could be interesting that whole genome sequencing is quite useful for. So as you've seen, they could be inverted, or they could jump much further. They could jump 100 kb down the chromosome, or even to a different chromosome. So whole genome sequencing allows you to check this. So in this case, the one I've shown, it was inserted downstream, but it's still too far away from any good candidate genes to have position effects, and the segregation didn't fit with the presumed inheritance pattern. So in this family there were no other CMVs of interest, so this family was another of these consanguineous ones. So I'm showing this slide. Again, it's the two affected individuals with array data. We're looking for shared homozygous regions, and then this is again the B-allele frequency plot from the whole genome sequence data here. So this is our candidate region that we're sort of still looking at. But the technical reason for sort of showing this slide is I'm just showing some controls at the bottom, and just sort of seeing how the clustering looks for different mean coverage whole genome sequencing data sets. So LDS10 was about 29x coverage along the chromosome. This control is 67x, and the bottom control is 17x. So you can see as you go from 17 to 29 and then to 67, this heterozygote cluster is getting much tighter. I mean, it's still not as good as the SNP array, but it's sort of pretty good once you get to this level. And once you get to this level, you can sort of start to pick up duplications as well, pretty effectively. So another thing we sort of played with is once we've developed a way of getting good B-allele frequency type plots from whole genome sequencing data. In some families where we've got two affected siblings, we thought sort of what happens if you just sort of do a sort of subtraction and sort of plot the absolute difference between your B-allele frequency score for each SNP along a chromosome, and that seems to be a really, really quick and effective way of picking out different IBD states. So if you had so this is real data for two affected siblings from the WGS 500 project, where I think this is showing chromosome 1, and we thought there would be a recessive mutation in this family. So the regions that are shared IBD2 that the software picks out quite nicely are the candidate regions. So we found that a lot quicker than like trying to reformat, like clean the data, format it for Merlin and running Merlin. We thought this was quite a nice sort of application. And then this is just showing exactly the same thing but from exome sequencing data. And although it's not quite as clean, it's still reasonably clear. So again, this is real data where we've got 3 affected kids, and we think it's probably recessive mechanism. So your candidate regions are where both parental chromosomes are shared in all 3. So theoretically, you should be able to reduce it to about 1/16 of the genome. And doing this sort of subtraction of the allele frequency scores, again, this is showing chromosome 1, we can sort of very quickly pick out the IBD sharing regions. So that's where I wanted to finish, and so all 3 cases that I talked about were published, so I thought this was a quick way of doing my acknowledgement slides, just because otherwise I was worried I was going to miss somebody. But also, if there's any questions, you can either refer to those you can read those papers for yourself. I also wanted to point out the two students who've worked a lot, the hardest on these projects. For case 2, it was Malcolm Howard, and for the third case, Hilary Martin did a lot of work on this and was the first author on the paper.